Alright, in this set of notes we're going to briefly discuss the structure of the cell membrane and then a quick review of protein synthesis from Chapter 4. Alright, the plasma membrane. This is the photograph from your book. The plasma membrane is made up of phospholipids, and here's what it might actually look like, and here's the representation. So it's that glycerol head and two fatty acid chains. So this is one of the macromolecules that we've talked about, and it falls in the lipid category. So the plasma membrane is made of phospholipids. It's a bilayer, meaning it's two layers of these phospholipids, and their little tails all point towards the center, and the glycerin heads are pointed towards the inside of the cell and the outside of the cell. You can call them phospholipids. You can call this a plasma membrane, the cell membrane. Both work. So there are a couple of unique features of eukaryotic cell membranes. They actually contain other lipids. One of them is called cholesterol. It helps make the membrane firm. So again, you've got a lipid, which is cholesterol, adding structure. It also keeps our cells from freezing in lower temperatures. So that's convenient. So this is the figure from your book. All of the little white or cream-colored circles with their little tails are the phospholipids. And there are lots of different macromolecules embedded here in the cell membrane. In blue, it's showing you some lipids. These are the cholesterol molecules. It's showing you some carbohydrates. So here's a carbohydrate portion that's attached to a protein. These are cell surface markers, and they identify what type of cell this is, kind of like a flag would identify what country. You also have proteins embedded in the cell membrane, and these are extremely important, especially for the cell transport. So things like osmosis and diffusion, these become important. So transport proteins help move substances across the cell membrane that can't just diffuse right through. So there are lots of things going on in the cell membrane. And the way we would describe the cell membrane as a fluid mosaic model. All of those little bits and pieces are just kind of freely floating around. If you imagine a lake that's packed with lily pads, and if you were to wade through those lily pads, the lily pads would represent perhaps the glycerol heads of the phospholipids, and your ankles or calves or part of your leg that's wading through those lily pads would represent the proteins that kind of move and swim freely through the phospholipid bilayer. They're not anchored down. A quick review of protein synthesis. It starts in the nucleus, where the DNA creates a messenger RNA strand. The messenger RNA strand leaves the nucleus, and it goes to the ribosomes, which are attached to the rough ER, or free-floating in the cytoplasm. And at the ribosome, the messenger RNA is used to create a chain of amino acids. That chain of amino acids is called a polypeptide, and this polypeptide chain is then folded and prepared in the endoplasmic reticulum, shown here, or in the Golgi apparatus, and some of them don't need extra folding or preparation at all. From the Golgi apparatus, they can be shipped to other parts of the cell. Pretty quick.